Hello, and welcome to Field Lab Earth, the podcast that's all about past and present advances in the fields of agronomic, crop, soil, and environmental sciences. Today we'll be talking to Dr. Merrin Macrae about phosphorus release from cover crops over winter conditions. What benefits do cover crops provide over winter? What relationship do they have with phosphorus runoff? And what kind of environmental trade-offs, if any, can result from their use? Answers to all these and more coming right up. I'm your host, Abby Morrison. Let's talk about science. Hi everybody, welcome to the show today. Today we have with us Merrin Macrae. Merrin did her undergraduate in environmental studies and her master's in geography at York University in Toronto, and her PhD at Wilfrid Laurier University with the Department of Geography and Environmental Management in Waterloo. She's worked in the Department of Geography and Environmental Management at the University of Waterloo since 2006. Hi Merrin, welcome to the show. How are you doing today?
I'm great. Thanks for having me on.
So happy to have you. So today we are talking about some relationships between phosphorus and cover crops. We actually have two episodes on both of those topics, but we've got our cover crops, plants that you're not really growing for making money or production. And then obviously phosphorus is a very important element for plant life. So can you tell us in general, what is the central question of your research? What is the relationship between cover crops and phosphorus that you were hoping to investigate with your research?
We're trying to develop and test solutions to reduce phosphorus losses from agricultural fields. And cover crops put on fields during the non-growing season are one conservation practice that's being recommended. And what the cover crops are doing is they're reducing surface erosion from the field, so that keeps the soil and the phosphorus bound to that soil on the fields rather than having it run off into the tributaries and into the lakes. The problem is that in some northern climates, they've actually found that cover crops can freeze and die in the wintertime. And when this happens, they release a lot of dissolved phosphorus to runoff. And so what this creates is a trade-off, and it can actually mean that you're not really solving the problem. And so this is something that's been shown in a lot of northern climates. But in the Great Lakes region, we are in a temperate region, so we do tend to get gentler frosts. And not only do we get gentler frosts in some of these more severe northern climates, we also have a lot of snow cover. And because of that, it tends to insulate our plants and our soil. And so the plants aren't necessarily exposed to the same level of freezing. So what we wanted to know was whether or not the cover crops in the Great Lakes region were actually releasing substantial amounts of phosphorus to runoff in the wintertime, and if this depended on the species that you were growing.
Yeah, and so you had some very specific objectives and questions that you were asking to try and answer this larger question. So can you tell me what some of the goals were of your project?
Sure. So what we were looking at is cover crops in a field setting, so not in a laboratory experiment, but out in the field. And what we wanted to know was whether or not cover crops released phosphorus following freeze-thaw cycles or winter, basically typical winter conditions. Did they release phosphorus? And if so, did this depend on the species, so different types of cover crop species that are typical of the ones that are grown in this region? Not only that, we wanted to know when was that phosphorus being released. So was it following the first frost in the fall, or was it really something that tended to happen in the peak of the winter or more towards snowmelt? And then finally, if we in fact saw that the plants were releasing phosphorus following these freeze-thaw cycles, did this actually translate to increased concentrations of phosphorus in the runoff water?
And can you describe how you set up your experiment to answer these questions?
Sure. So we did this all on a single field that we divided up into a series of plots. So they were about 1 acre each. And each of the plots that we used had a different cover crop species or species mixture grown. And then we left one of our plots without any cover crops on it as a control. And so within these, the farmer that we were working with, he planted the cover crops in August. And so what we then had was a series of runoff collectors that we installed in the fields that would allow us to capture water samples after a rain or a snowmelt event. And so the runoff collectors, some of them captured surface runoff, so water above the ground, and some of them captured water moving below the ground in the very shallow subsurface and a little bit deeper down. And so what we did is we monitored the situation from August all the way through April. So as the fall carried forward and we started to have some fall rains and some early freeze-thaw cycles, we would go out and collect soil and vegetation or cover crop samples and bring them back to the laboratory and measure how much phosphorus, extractable phosphorus, that they had on them. And at the same time, we would take surface runoff samples and subsurface water samples if we'd had a runoff event.
And can you describe the different kind of runoff collectors that we're using, both above and below ground?
Sure. So what we used above the ground is we used a series of weirs where we had edging that was installed above the ground that would concentrate the overland flow into a V, into a narrow opening. And then we captured that water in a container that was installed. It was buried just a little bit below the ground, but the opening was flush with the surface. And so it would direct that surface runoff right into the receptacle that we had to collect the water. And we actually used Nalgene bottles, and what they had is they had a hole drilled in the lid, and then you have a ping pong ball that you put inside the Nalgene bottle. And what happens is that when the water fills that bottle, the ping pong ball it's going to float on the water. And so what it does is it actually plugs the hole in the top of the sample bottle so that your water sample doesn't get contaminated until you can go out there and retrieve that sample. And then below the ground, we used something that we often use in hydrology research. It's called a piezometer. And what it is it's a pipe that is open to the atmosphere, and it's vertical above the ground, and then it's installed below the ground, and we have what's called a screening depth. And that's essentially the depth in the soil that is slotted. So there are basically holes drilled in that pipe. And so what that will do is it will give you a water sample, and it'll give you additional information as well about, it can tell you where the water is flowing as well. But for our purposes, it allowed us to get a sample from, say, 0 to 5 centimeters, and then maybe 5 to 10 centimeters, 10 to 15 centimeters, and so on.
I love this because I just think the things that scientists come up with to engineer their way into getting the samples they need is so fascinating to me, and I'm always amazed. And it's so simple. Like with the Nalgene bottle, I love that. And I'm so glad you explained how the ping pong ball worked, because when I read your paper, I was like, how do they get the ping pong ball inside? And then I was like, oh, it's because the lid is bigger.
Yeah.
I got it. I got it eventually.
Well, I didn't come up with that method. I think either USGS or USDA, somebody published a paper long ago using that, and I absolutely loved it. So as a method, and so it was great to be able to use it here.
Oh my gosh, that's wonderful. If anyone knows that paper, feel free to shout out on Twitter and let us know. We would love to give them a cyber shout out as well. At Field Lab Earth is our Twitter. So, yeah, you also then were taking samples of the plants themselves throughout this time period. Is that correct?
Yes, actually the plants and the soil.
And how did you go about doing that?
So we we really just left the plants growing. So we didn't do anything to it. We didn't clip it. We didn't kill it. We just let it freeze up green, just the way that people normally would. And we simply just went out, and when we wanted to get a plant sample, we just clipped. I mean, you had to try and get representative samples. So you'd randomly collect, obviously more than one plant. You would collect several plants, and then you would just literally go out there with clippers and put it in a sterile baggie and bring it back into the lab. And then for soil, the same. We would just simply use a soil sampler and get a composite sample. So you get a bunch of subsamples of the soil, so that you take more than one sample so that you get something that is fairly representative of what it is that you're trying to sample. And then once we collected this material, we brought it back to the lab, and we analyzed it for what they call extractable or water extractable phosphorus.
Sure. And where exactly were your experimental sites again?
It was actually in Bloomingdale, which is just outside of Waterloo, Ontario.
All right. And how was it collecting those samples in winter? I mean, I think a lot of times we think of agricultural, or crop or soil science is usually more in the warmer months, where it's a little easier to get places or to collect samples. I mean, did you have to deal with, there was the one blizzard that was 3 feet of snow in 48 hours, or ice conditions that would make, or permafrost? I mean, what was that like?
Yeah, it's definitely challenging to sample things in winter. So this is the master's thesis work of James Cober, and he was an absolute trooper going out to collect this data throughout the non-growing season. And so, yeah, he absolutely had to deal with snowstorms and rain on snow events. And there were definitely times, there was at least one occasion where he couldn't get a soil sample because we had lost all of the snowpack, and then we'd had a freezing cycle, and the soils were frozen, and he just couldn't get into the soil to actually get a sample. And this is just one of those things you sometimes have to live with. It's just the nature of doing winter field work. But yeah, definitely trying to make sure that your instrumentation works and that things don't get frozen up or blocked, or your weather station is still working and you have the right battery power. It's definitely not easy in the winter, and it's obviously really, really cold at times. So you just dress warm and don't go out alone, and do your best. Yeah,
I readily admit, even as a homegrown Wisconsinite, that I would not enjoy doing this research. That is not for me, I think. Although, I mean, I guess you could have a snowball fight or make a little snow farmer or something in your field. That'd be pretty fun. Maybe not as scientific, but definitely fun. So something that I'm always curious about also in related to this is how you actually get the site that you were working on. You mentioned that this was, an actual farmer was working on this field. So how did that all shake out?
Well, this is actually a really interesting story. So we were looking to get a farm field, and we have individuals that we've been working with for some time that are maybe an hour or 2 or 3 hours from the university. But in this particular circumstance, we wanted to have a site that was very close to Waterloo that we could run out to fairly easily. So what we did is I put out a message on Twitter, and I had said in this message, in this tweet, that I was looking for somebody who would be willing to do a cover crop trial on their farm that was within an hour's drive of Waterloo. And I got 50 replies within 1 hour from people willing to volunteer their property to do this. And so it was really exciting because, well, first of all, we found a site and we found a partner to work with for this study. But second, it really showed me just how engaged the farming community is in this region and how interested in answers they are, and just how willing to support research they are.
I love that. I love every part of that story, from just using Twitter to make those connections. I think is always fun. We'll have, your Twitter account in our show notes. So if people want to connect with you, they certainly can. As well as, just that engagement is so lovely to hear about, and getting those connections is super important, obviously, but also just really fun and neat. Hi, everyone. I hope you're enjoying the show. Interested in learning more? Merrin's article, Winter Phosphorus Release from Cover Crops and Linkages with Runoff Chemistry, published in the Agricultural Water Quality in Cold Environments Special Section of the Journal of Environmental Quality, is always freely available. You can find a link to it in our show notes. Let's get back to the show. So you did your experiment from August until, where was the cutoff?
April. So you're getting your samples of your water, your soil, your crops, testing all the time. What did you find? What were your results?
Well, we found several things. First of all, we found that different species of cover crops are affected differently by frost. And so they released different amounts, and some released more than others. So things like oat or tillage radish, they released a fair amount, whereas things like vetch, hairy vetch, or cereal rye didn't release quite as much. And so the frost-tolerant species, things like vetch and cereal rye, they didn't release a lot of phosphorus after the freeze-thaw cycles, because our frosts are gentle enough that these plants can withstand them and survive over the winter, whereas the other ones are a little bit more tender and more vulnerable to frost kill. And so we know from a previous lab study that James Cober, the master student, did, that was published in Agronomy Journal, that if even those hardy plants get a severe frost, like the ones that are experienced, say, in the northern Great Plains or in some of the Nordic countries, even those hardy plants will release phosphorus because they will obviously be killed once you get a severe enough frost. And so in our field study, we found that yes, plants can release phosphorus following freeze-thaw cycles, but some plants are better than others. So the second thing that we found is that even though the plants can release phosphorus following freeze-thaw cycles, we actually found that the soil was holding as much phosphorus or more than the plant material in a field. So if I take a gram of plant material and I take a gram of soil material, the plants have a lot more phosphorus in them per gram that they can release as a concentration, meaning per gram that they can release. But because there is so much soil in that field, so the density of that soil compared to, say, the biomass of the plant matter, there's so much soil in that field that there is way more phosphorus being held even in the top inch or two of the soil compared to all the phosphorus that's being held in those plants. And so the soil was a much bigger source of phosphorus, comparatively speaking, compared to, say, the plants. And then the third thing that we found is that even though both the plants and the soil can release phosphorus, what we found is that the phosphorus concentrations in water, so that would be your surface runoff and your shallow groundwater, the subsurface water, we found that the concentrations in water tended to resemble the soil phosphorus rather than the plant phosphorus. So the differences in the phosphorus released by the plants following these freeze-thaw cycles did not translate to differences in concentrations of phosphorus and runoff, and the soil really regulated how much phosphorus was either going to be held or released by the field.
So there's actually another challenge that you guys face that I wanted to ask you about, and it has to do with the surface runoff that you were trying to collect. Can you just talk about some of the things you have to wrestle with an experiment like this with surface runoff?
Sure. So the one thing about surface runoff is that it is something that, although it does occur, it doesn't occur very often. It tends to only occur following peak flow events. So because of that, you don't always get a lot of opportunities to sample surface runoff.
But secondly, surface runoff isn't something that is going to be generated exactly the same from each of the plots within a given field. And instead, it's going to tend to follow the topography of the field. And so you try to orient your plots or lay them out in a way where you're going to be able to capture some of that surface runoff, and you do the best that you can. But inevitably, not all of the plots are going to supply surface runoff, or they may not do it as often. And this is just a challenge with a natural field and trying to do things at a plot scale within a field. You may not get the exact same hydrologic response from every one of the plots. So an alternative approach is to simply have adjacent fields and have different maybe plants, or basically use side-by-side fields to try to compare things that way. The problem with side-by-side fields is that you may still have differences between those side-by-side fields that are natural that can exceed your treatment effects. So it's just one of these challenges that we always face in fieldwork. So obviously more studies, more opportunities, longer time period to capture more and more of these events is obviously going to give you more and more confidence.
Yeah, absolutely. I think that's so interesting just to think about as a challenge, or, you know, what math hoops you might have to jump through to try to overcome some of those differences. And then another couple of sub-challenges that I wanted to ask about. Because you were doing this in winter, did you also face, like, maybe a runoff event was going to start, but then it, like, froze, and all your runoff is just now frozen on the side of a small ridge or something? And then also, with your ping pong ball Nalgene bottles, were you worried about, like, oh, it snowed a bunch, so now we've got all this just snow water in these, or was that not really a factor because I mean, any thoughts on that?
Okay, so there are some areas where you can get a lot of melt and refreeze, and then you do have this frozen ridge. I know that's something that they have to deal with in really cold areas like the northern Great Plains, and we've done some field work out there and we've seen that happen, and that can be really tricky. We didn't really see that. What we often saw is we would see a freeze-thaw, but we really didn't necessarily ever see a condition where the temperatures plummeted while you still had standing water that would cause it to freeze. It's just not the nature of how things happen around here. And again, I think that tends to be because we have a more moderate climate in this region, and just the nature of the types of systems that went through. And so certainly we had times where we would have a mixture of precipitation. So we would have snow, and then it would melt, and then we would have rain on snow, and it would be just a big mess of icy, snowy runoff water that would be flowing over the surface and into those Nalgene bottles. But in fact, we sampled that anyway because that is indicative of what the conditions are. So we didn't try to only get rainfall runoff events or snowmelt runoff. We just captured every runoff event to see what types of concentrations we might observe and if they differed between the events throughout time and with the different species in the field.
Sure. And then something I forgot to ask about earlier, you had briefly touched on this, is the cover crops that you used. Can you tell me a little bit about kind of how you decided what cover crops or cover crop mixes you were going to use for this experiment?
So we picked the species that we did based on the types of cover crops that are prevalent in this region. And so one of my collaborators in this work, Laura Van Aerd of the University of Guelph, works a lot with cover crops. And so we had consultations between her and myself and James Cober, the master's student, and we chose a range of crop types that tend to be most typical of this region. And two other individuals who also consulted with us on that were OMAFRA staff members, Anne Verhollen and Adam Hayes, who also work extensively with cover crops and farmers in the area. And so we just took a range of varieties that people are working with, and we also made sure that from that we had ones that tended to be hardier and other ones that tended to be more tender. And then we also had a multi-species mix. So we wanted to see if monocultures, meaning just single crops themselves, behave differently than, say, the multi-species mixes.
Yeah, that's awesome. It's always fun to hear how people choose the different parameters that they test. So now we've kind of talked about what you found out and what you tested. On a grander scale, why is it so important to do this kind of research? Like, how is this helping the day-to-day researcher, farmer, general public, human? Why is it so crucial that we do research like this now?
So we're really trying to reduce phosphorus losses from agricultural fields. Farmers don't want to have their nutrients running off of their fields, and we don't want to have phosphorus running into the rivers and the lakes. So we're really trying to come up with strategies that are going to work to keep that phosphorus on the fields so that it's there where the crops need it the following season. So farmers are being given a lot of options as conservation practices for things that they can do. And so cover crops are not just important for phosphorus, they're also important for soil health and nutrients, managing nutrients on the fields, for example, your nitrogen. And so they really are something that are being recommended as a conservation practice. And they're also good for weed control. The problem is, like I mentioned before, there are some studies that are saying that cover crops can actually make the problem worse when they freeze and release phosphorus in winter. So what ends up happening is that people are being told, do this, don't do this, and it gets very, very confusing. And so what we were trying to do is try to tease out where are cover crops more and less effective, and where is that risk of winter loss with snowmelt greater? And what we found is that the farmers can have the benefits of the cover crop without those winter trade-offs because of the snow and the gentler frosts that we have in the region that can protect the plants from those severe frosts. So it means that really cover crops within the Great Lakes region, at least certainly where our field site was, so this is sort of in the Waterloo region, but we're finding that, yeah, cover crops are probably a really good idea here because you don't have that substantial risk. But other areas, say in Winnipeg, they have to be a lot more cautious with the use of cover crops because we know that when things freeze out there, you can have problems. So I think it's important that we do a better job of helping people make informed decisions so that they're using the right practice in the right place. So if cover crops are going to have a net benefit here, then great, we should use them. If, on the other hand, they were not going to have a net benefit, then we shouldn't be emphasizing them. So we're finding that there really seems to be a benefit.
Absolutely. Yeah, that's really, really important stuff. Which kind of leads us, we've touched on this a little bit, is part of that whole process is doing, more research projects to, tease out more of those differences between the runoff or location and temperature and all that kind of thing. What else do you see on the horizon for research in this area?
So cover crops are one of the conservation practices that we've been exploring, but we've also looked at 4R's nutrient management strategies, so things like subsurface placement or the incorporation of manure following the surface broadcast in the autumn. So we've looked at a range of different types of conservation practices and how they're actually performing under winter conditions. And so some of the work that we've done is just trying to understand or identify when most of our water and our phosphorus are leaving the fields, how they're leaving the fields, and what we can do about it in terms of conservation practices that are going to really work effectively during those times of year. And so a lot of the conservation practices that we historically emphasized were ones that were developed during the growing season when things were growing, things like lush plants like grassed waterways and riparian buffer strips and things like that. And so those are still really great things that you can do, but you can't necessarily rely on them in March when everything is maybe encased in ice or not growing. And so we're looking at individual and combinations of conservation practices that we can use to try to minimize phosphorus losses. And tillage and no-till is another one that we've looked at.
Sure. Yeah, that's all really important work. Thank you for doing that. I have 3 questions left for you. The first one is, if listeners want to learn more about these topics, where can they go for more information?
People can learn more about cover crops in cold regions in the article that we're talking about today, but also from several other articles that have been recently published from our lab and our team over the last couple of years. And so the thesis work of James Cober and Tatianna Lozier were both published, and so there's about 4 papers that involve both field and lab studies that were looking at how cover crops responded to freeze-thaw cycles in winter. And I was recently lucky enough to collaborate with Jean Liu from the University of Saskatchewan, where Jean led a review article that was looking at cover crops under cold climates. And so that was published in the special issue of the Journal of Environmental Quality. So there have been other groups at the University of Manitoba and with Environment Canada and the University of Saskatchewan who have also done significant work looking at cover crops and how they function in cold climates. And there's also been a lot of really great work written over in Europe and in the Nordic countries as well. People can also learn more about the Agricultural Water Futures Project. In this project, we're trying to understand how climate change will affect water supply and water quality, and also socioeconomic drivers of farmer decision making. And so a big part of this project is understanding cold region processes and figuring out which management practices work under cold conditions. And so the special section that the current paper was published in is Agricultural Water Quality in Cold Conditions. And this was led by a smaller portion of the entire Agricultural Water Futures team. So Jean Liu, who's a postdoc based out of the University of Saskatchewan, Helen Balch, Jane Elliott, Henry Wilson, and myself, in addition to some other guest editors, we led this special section to try to have a consensus and an improved understanding, not just in North America, but globally, on how management practices or different conservation practices work under cold conditions.
Wonderful. We'll include those in the show notes. Next question is, if people want to get involved in this research, the conservation practices, anything related to this, how can they get involved?
Well, I think, first of all, I would like to encourage people to try cover crops. Even if they don't put it on all of their fields or a complete field, try it on a section of a field. And certainly you can talk to your conservation authorities or your crop advisors to look at how to do this the best way and learn a little bit more about it. But I would encourage people to try cover crops and see if they like the benefit that it has for their crops and see if they can visibly see an improvement to the water quality that's leaving their fields. The other thing I think people could help us with is to tell us what can be done to make this feasible for farmers, both financially or logistically, because we never want to be recommending things to people that they can't do or that don't make sense. So if there's a barrier or a reason why people can't do this, communicating with us about that to make it possible for them to try would also be a step in the right direction.
Wonderful. That's some great advice. I hope people take it. And then final question for you is, what is one fun fact that listeners would not know about you if all they had was your research?
Well, I'm a wife and a mother of 2 kids. I have a daughter and a son. I would definitely say that parenting takes up a substantial amount of my time when I'm not at work, but that's just the stage of life that I'm in. But when I do get some free time, I'm learning to knit thanks to YouTube. And sometimes I post photos of the things that I knit in my Twitter feed.
Wonderful. Do you have examples of things that you've recently knit?
Yeah, I actually did my first sweater. and it was stranding or fair isle knitting, and I made it for my son, and I did it actually while I was over in Switzerland, so it was pretty exciting. It was in July, and my son was so excited to get the sweater that he tried it on, even though it was about 33 degrees out.
Oh. Well, that is so wonderful. I'm really happy to hear that story. So, yeah, that's all we have for you today. But thank you so much for being on the show. I loved learning all about your research, and thank you for doing it.
Well, thank you so much for a great conversation. It's been a pleasure.
Wonderful. Thank you for listening to Field Lab Earth. You'll find a link to today's paper and other resources for this episode in our show notes or on our website. If you have any questions, comments, or recommendations for show topics, please contact us at podcast@sciencesocieties.org or on Twitter @FieldLabEarth. If you'd like to hear more content like this, please subscribe, and don't forget to rate and review us on Apple Podcasts or anywhere else you find your podcast if you like our show. This podcast is a joint production of the American Society of Agronomy, Crop Science Society of America, and Soil Science Society of America. Special thanks to Lobo Loco for the use of their song Spook Castle on the intro and outro of our show. Opinions and conclusions expressed by authors are their own and are not considered as those of the American Society of Agronomy, Crop Science Society of America, Soil Science Society of America, its staff, its members, or its advertisers.